Good morning students. Today we are going to discuss about the application of the recombinant DNA. So in the last two videos we have already discussed about the genetic engineering, what is recombinant DNA and what are the tools used for the recombinant DNA, as well as what are the steps which are involved in the genetic engineering or recombinant DNA technology. So today we are going to see the applications of the genetic engineering or recombinant DNA. So there are many applications under this recombinant DNA technology, and it is you can say that this is the one of the very important method of recombinant DNA which is applied now in many fields. First one is the gene therapy, second one is the production of transgenic plants. So in order to produce a new plant or transgenic plants or resistant variety plants, disease resistant variety plants, we can apply this, that is genetic engineering field. So it is one of the applied field. So it is also applied in the application, you can see in the production of transgenic animal, study of genes, diagnosis of the diseases, identification of the parents, nitrogen fixations, production of antibodies and vaccines, and productions of protein and environmental biotechnology. So we are going to discuss all this application one by one. So the first step, that is said to be the gene therapy. So what is a gene therapy? So gene therapy, it holds the potential for the cure for many human diseases, genetic disorders. So such as, you know that one of the genetic disorders such as hemophilia or the sickle cell anemia or color blindness, yeah, L-capton urea. So these are the certain genetically inherited diseases which transfer from one parents to the another parents. So this can be rectified by means of the gene therapy. So you have already heard of the hemophilia. What is hemophilia? Hemophilia is one of the mostly inherited genetic disorder. एक ऐसा प्रकार का inherited genetic disorder है in which the blood doesn't clot. So here you can find out in this image, the blood doesn't clot or clot formation doesn't takes place, and because of this excessive bleeding occurs after many or any type of injury or damage. So this type of defects can be rectified by means of the gene therapy. So you have here you can find out a picture in which the hemophilia is seen, where the clot formation doesn't take place and excessive bleeding sometimes may cause the death. Next is the next image is related to the, this is L-capton urea related images. So here it is a inherited conditions that in this L-captonuria you can say that the urine used to normally turn black when exposed to the air. So if a person is suffering from L-captonuria, their urine normally turns black when it is exposed to the air. And this is the normal person urine, which almost remains yellow in color. But a person, if he is suffering from L-captonuria, then their urine normally, when it comes in contact with the air, it turns black. So gene therapy, you can say that it holds the potential for cure for many human genetic disorders. And these may be hemophilia, alkaptonuria, color blindness also. So how these diseases can be cured? By replacement of defective genes. So the defective genes which are responsible for this type of disorders or diseases, that can be replaced. जब हम इस प्रकार के डिफेक्टेड पेज को, अगर डिफेक्टेड जीन्स को रिप्लेस करते हैं by means of replacement of the defective gene by the normal healthy gene. This can be rectified. Next application is the production of transgenic plants. So by using the genetic engineering techniques, transgenic plants with the desired characters can be produced. हमें जो सा whatever character which we need, that is desired characters that can be introduced into the plants. And this technique involves introduction of the required gene. हम क्या करते हैं, एक required हमारे interested gene को हम क्या करते हैं, introduce करते हैं. और जीन फ्रॉम दी अदर ऑर्गेनिज्म में से हमारे डिजायर्ड जीन्स हम क्या करते हैं, पहले आइसोलेट करते हैं एंड देन वी आर गोइंग टू रिप्लेस इट, एंड इन दिस वे वी कैन गेट अ न्यू वेराइटी. एग्जाम्पल फॉर गोल्डन राइस. सो गोल्डन राइस यू कैन से इट इज वन ऑफ द ट्रांसजेनिक प्लांट. So here in this picture you can find out the picture of a golden rice. And now why this golden rice is produced? So you can say that the golden rice is a variety of rice, एक ऐसा variety of rice है which is produced through genetic engineering. genetic engineering through इसे produce किया गया है. Why? To biosynthesize the beta carotene. So in order to synthesize the beta carotene, a precursor of vitamin A. We can say that this is the source of the vitamin A. So in order to get the vitamin A, this type of or this variety of the plants or the Oryza sativa is genetic engineering has been done on the Oryza sativa in order to get the large amount of vitamin A. So it is one of the edible part of the rice which is normally utilized, and it is the source of the beta carotene. So in this image you can see this is nothing but the Bt cotton, Bt. So it is again a genetically modified organism, or it is one of the genetically modified plant, or you can say that this, what is the specialty of this plant is that it is genetically modified pest resistant plant. So this cotton plant is pest resistant, it doesn't get the attack of pest, and hence the production is the highest. So you can say that this type of variety can be obtained by means of the genetic engineering. So you can say that Bt cotton is genetically modified organism or genetically modified pest resistant plant, cotton variety, and which produces an insecticide to combat the bollworm. Here in this Bt cotton, it is an insect or pest resistant plant. You can say that it is one of the plant which is produced by means of recombinant DNA और genetically engineering plant है, जिसमें कि these plants they can resist the pest. So you can say that they are the pest resistant plant, pest resistant plant, so that their production can be achieved on the large scale. And the Bt stands for the Bacillus thuringiensis. So it is one of the insect resistant transgenic crop designed to combat the bollworm. Bollworm से resistant variety होने के कारण it can give more and more production. Next, coming up to the production of transgenic animal. So the transgenic animal with the desired character can be produced. हमें जो character चाहिए उस type of character can be produced. For example, here in case of picture you can say that you can see the sheep. So it is a picture of a sheep which is produces more wool, or more wool can be produced by means of the transgenic animal, or by means of transgenic you can say that genetic engineering. So it is again a modified animal sheep. On sheep certain genetic engineering has been done, and after that genetic engineering that interested desired gene can produce more amount of the wool. So here we can say that more production of wool can be extracted from this type of sheep, and it can be also engineered on the goat, which produces a large amount of protein having the capacity to dissolve the blood clot. So the production of the transgenic animal can also be possible. It is again one of the application of the genetic engineering. Next is study of the gene. So if the defected gene can be studied under the genetic engineering, here one can study the defected gene. So you can say that defected genes can be studied under the genetic engineering for the improvement of health. So if a person contain any type of defected gene, that can be removed and replaced, and so that a person can be converted into a healthy human. Next is the diagnosis of the diseases. So here one more point it can be added, that is the diagnosis of diseases. Here the genetic disorders and also infectious diseases can be identified. हम इसमें क्या करते हैं, जो भी infectious diseases है उसे identify कर सकते हैं, and by using the different techniques of genetic engineering. So if a person is suffering from any type of infectious diseases, that can be identified and that can be diagnosed. So the study of the genes and the diagnosis of the disease both can be done by means of, or you can say that this is the application of the genetic engineering or recombinant DNA. Next is the identification of the parents. So if in case of dispute parentage, मतलब if the offspring belongs to which parent, if there is any type of dispute, in this case the DNA fingerprinting can be of the immense use. तो वहाँ पे DNA fingerprinting करके हम find out कर सकते हैं who is the correct parent of the given offspring. So next one is the nitrogen fixation. So you know that the NIF genes, NIF, nitrogen fixation genes. NIF stands for nitrogen fixation genes. So NIF genes, these are responsible for nitrogen fixation, and that can be introduced into a plant directly so that the plant can fix the nitrogen on their own. So you know that only the leguminous plants, they can fix the nitrogen. What is the main function of this rhizobium bacteria which is present? So almost these leguminous plants, they contain the inside the root, they contains the root nodules. So in these plants, leguminous plants, they contain the root nodules inside the root. So you can find out these are the root nodules. Inside the root nodules, rhizobium bacteria is present. What is the main function of rhizobium bacteria is that they used to live in the root of the leguminous plants, and they used to fix the nitrogen, atmospheric nitrogen. So you know that 78% of the nitrogen which is present in the atmosphere, they had got the capability to convert this nitrogen into the nitrate form. So they convert the atmospheric nitrogen in the nitrate form, and this is the form in which the plant they utilize the nitrogen. So it makes the available nitrogen to the plant. So if the nitrogen fixing genes which is present in the leguminous leguminous plant, if nitrogen fixing genes, if it is present normally in the rhizobium bacteria, that can be replaced to a normal plant containing this type of gene, then it may be possible that the plant may synthesize, or the plant may convert the may convert the nitrogen into nitrate form. So this type of nitrogen fixing can be achieved in the non-leguminous plant also. So this technique is again applicable in the nitrogen fixation. Next is the production of antibiotics and the vaccine. So many viral vaccines and bacterial antibiotics, they can be produced by using the various genetic engineering techniques. तो बहुत प्रकार के वायरल वैक्सीन है या फिर बैक्टीरियल एंटीबायोटिक्स है हम अचीव कर सकते हैं by means of this technique. Next is the production of antibiotics, production of proteins. Next is the production of proteins. So in many mammalians, proteins which are the essential for normal health of the human, example for insulin. So insulin is one of the protein that can be produced by using the proper microorganism. तो इंसुलिन हम क्या कर सकते हैं, प्रोड्यूस कर सकते हैं by means of a proper microorganism. So thus there are certain microorganisms which produces the insulin. So if we culture those organisms and then if we take that part of the protein producing part of the gene, then this production of protein can be possible. Next is the environmental biotechnology. So the last point under the application is the environmental biotechnology. Here the gene for the biodegradation of many toxic waste. So toxic waste जो भी है हमारे पास, so in order to remove the toxic waste or biodegradation process में, we can say that in case of wastewater pollution, can this type of particular type of gene can be synthesized for the purpose of the environmental cleanup. So if we want to clean the environment, in order to environment cleanup, certain genes of biodegradation, genes of biodegradation of many toxic waste and wastewater pollutants can be synthesized. And you know that genetic engineering provides a vast opportunity for improving the process of economic value and in treating the diseases. हम यहाँ क्या कर सकते हैं, by means of genetic engineering what we are doing, we are improving the process of economic value. यहाँ पे BT cotton हम produce करके हम क्या करते हैं, economic value को increase कर रहे हैं. At the same time, in order to find out, first we are going to diagnose the disease को हम diagnose भी कर सकते हैं, we can rectify the disease also. Even the medicines, agriculture, industry and are finding important use of cloning and gene expression technology. इस टेक्नोलॉजी में हम हर एक फील्ड में, मेडिसिन की फील्ड हो, एग्रीकल्चर का फील्ड हो, इंडस्ट्री का फील्ड है, सब जगह इसका यूटिलाइज कर सकते हैं. सो इन एडिशन टू जेनेटिक इंजीनियरिंग हैज revolutionized genetic studies in higher organism and in powerful tools in basic research on microbiology, microbial genetics. So आज के वर्ल्ड में it is the one of the very important and powerful tools. It is one of the important and powerful tools in the research and microbial genetics. Powerful tools in microbial genetics. So today we have seen what are the different application of the genetic engineering या फिर recombinant DNA technology. So what are the applications? We have got almost 10 applications. First one is the gene therapy. Second is the production in transgenic plant. Third is the production of transgenic animal. Study of genes, diagnosis of diseases, identification of parents, nitrogen fixation, production of antibodies and vaccine, production of protein and environmental biotechnology. So for today we stop here the topic. See you tomorrow.